assume that I do that.
No, I'm not going to talk about any of that.
We evolved beyond it. We have evolved beyond it.
So in the last couple of minutes, I decided that, actually half an hour or so, I decided that this is really a tough room. It's more shaped like a bowling alley than a lecture room. This is a tough crowd. There are some things, in preparing this.
Bring your mic up close up, bring your mic up.
See what I mean? Okay. I didn't. Gotcha. I had no idea who would be here, and I did know that there'd be various interests in educational background and so forth. I knew that not all of you would be biologists, if any of you are biologists. And therefore, and I knew that if I simply asserted things about genetics to you, you guys are not going to take it on faith, what I said. So I decided to give a rather general talk, introducing you to the cave environment, cave fishes, talk about the remarkable resemblance between unrelated species all around the world living in the same environment. It's something that's called evolutionary convergence. And so these guys here are the bread and butter of my laboratory. These are Mexican tetras. There are surface forms and cave forms there. Does everybody know which is the surface form and which is the cave form? Okay, we're on the same page for that. The beauty of this system is that these guys, while ancestral to these guys, and separated by several million years of evolution, are still close enough that they can interbreed. So I can actually do genetic studies. I can cross the cave fish with the surface fish, make hybrids. The hybrids are intermediate. I take an F1 hybrid, which had a cave and surface parent, and I cross it to another F1 hybrid. I get an F2. And for those of you who remember your high school or your college genetics, or biology, you get this 1 to 2 to 1 ratio of cave homozygote, heterozygote cave surface, and surface homozygote. And that permits me to do genetic studies on these guys. Somehow, I think I can do that from here. No, I can't.
Yes, you can. Yes, you can.
So the differences between them reflect these very different environments in which they live.
Escape.
No, escape kills the whole thing. And so basically, life on the surface of Earth and life below Earth. On the surface of Earth, at least you have light 12 hours a day, and below Earth it's perpetual darkness. And the secondary effect of this is that food is plentiful, or relatively plentiful, on the surface of the Earth because you have photosynthesis. In the cave, you don't have photosynthesis. All cave ecosystems are very much attenuated because the energy input at the base is very low. Cave fish are generally the top predators. Little rinky-dinky things that can be as small as my pinky. They don't have to fear predators. There's not enough energy there to support a predator population, and generally speaking, they don't have to deal very much with competition from other species. So it's a simplified environment. It looks formidable for us. For us to live in a cave would be extremely difficult, but it actually, in some ways, is a comforting haven for cave fish. So what I decided to do today was to talk about caves in general, the cave environment, cave fish, illustrate what I mentioned to you before, this evolution to resemble one another, which is called convergence, and make a point about convergence, which is that convergence is the same as an evolutionary replication. I'm an evolutionist. I like to tease my evolutionist colleagues by telling them that evolutionary biology is really lousy science. It's crummy science compared to laboratory science. Because in laboratory science, you have the replicative experiment. You have a hypothesis, you design an experiment to test the hypothesis, because the hypothesis has certain predictions. You perform your experiment, It either rejects the results reject the hypothesis, or conditionally you accept it, you modify the experiment, blah, blah, blah. You can do a tremendous amount with that approach. So we already know the answer to the first of them, but who are the two great biologists? Oh, and the other thing about this room is that those guys are downstairs, but that's okay. Who are the two great biologists of the 19th century? Charles Darwin and
Weizmann?
Wallace.
Gregor Mendel? Is that
Huxley?
I guess there can be different opinions, but I would vote for those two as the top biologists of the 19th century. I was going to say the difference between downstairs and upstairs is that they're drunk and we're sober. Now I'm not so sure. I'm sorry, Pete. So
You do stand-up comedy too, don't you?
this is, everybody laughs at me, so natural born comic. So Mendel was a great experimentalist, and as I was discussing earlier, Mendel actually was trained as a physicist, and so he did these beautiful experiments with plants. And while Darwin was also a wonderful experimentalist and did marvelous work, we actually remember him for his observations and what he did with his observations. And this dichotomy, observationalist science in evolution, experimental science, and just about all other aspects of science, this dichotomy exists today. So there are lots of extremely important things that happened in the history of life. Insects, for example, developed a waterproof cuticle. And that waterproof cuticle permitted them to go all over the land. Just about every landmass that's not frozen perpetually has insects on it because they don't dry out. Birds develop feathered flight. The amniote egg was developed. Amphibians have to go back to water to breed, but reptiles, birds, and mammals don't have to. So extraordinarily important changes in the history of life, but these were events that happened only one time. And because they happened only one time, if you have any idea, any hypothesis about them, you have no independent data set to test your hypothesis. But with cavefish, there are 100 species of cavefish around the world that are known, probably many more to discover, and every one of them is a replicate of an experiment that asks what happens when you turn out the lights and limit the amount of food. So I think this aspect here, convergence equals evolutionary replication, is rather important. Darwin knew about cavefish, but was really puzzled and incorrect in his interpretation of the loss of their eyes. So we'll talk a little bit about Darwin's dilemma. I want to speak as well about how we can do genetic manipulations, simple hybridization of different cavefish populations and restore vision. So things that have come from populations that have been in the dark for 1 million years and probably for most of that 1 million years couldn't even see. In one generation of hybridization we can make hybrids that do see. And then if we get to it, and because I was told 30 or 40 minutes, maybe we won't get to it, we're going to talk about natural selection against eyes and also sleep. One of the things about cavefish that we've discovered is they don't really sleep very much. So this shows the entrance to a cave in northwest Thailand. I'm one of these little specks down here. It's an extraordinary through cave. The river goes in one side of the mountain, comes out the other side of the mountain. This is the side that it comes out. I've been there about 3 or 4 times, and every time I go there I end up sleeping exhausted on that rock at night, because to walk down from that breakdown pile is dangerous. It's odd that it should be dangerous to walk down a pile of rocks at night if you've just been caving, but somehow we feel that. This is in the cave, some distance away, looking back at that big pile of rocks there. And all caves, of course, have entrances, and in the entrance is light. If the cave is really a short run through a mountain, then you really don't have blind cave fishes, and it has to be a longer run through the mountain. This is a cave that's only about 1 kilometer long from this entrance to the exit. We looked for cave fish there, again in northwest Thailand, found lots of surface fish though. Not every cave can be walked into, and so this is Vasquez Cave, a map of Vasquez in Mexico. The fish I wanted are down here, that's 915 feet down, and you do that on ropes. It will not be very effective to play it this way. It just shows my former graduate student Luis setting ropes in one of the caves there, and there was a soundtrack here, which I don't think we'll hear. Had I known he was going to do this, I would not have permitted him to do this.
What's he doing? Wow.
He's setting the rope, and what he says now is rope down. And what you do to get those fish, and these are the Mexican fish that I work on, you simply rappel into the cave and climb up. It's the climbing up that's the hard part. Without a soundtrack, there's probably little point in playing this further. That's my present graduate student, Martina Bradic. She's rappelling into that same cave a number of years later, getting set to rappel in. She had absolutely no experience at doing this. We trained her. That's ain't gonna work anyway. Thank you. So we trained her. This was the first pitch she dropped, 68 meters on a half-inch thick rope. And to give us some confidence, what we did is we did some politicking in town and got the roof of the fire department to practice rappelling techniques. This is a flowstone formation in Melana Cave in Thailand. The riverbed is here. The main river flows through the mountain from one side to the other, but there are internal tributaries that leave a flowstone deposit. And on the top, in the gores, little holes on top, you find one species of cavefish, and actually over here, in the water that's dripping off, there's another species of cavefish, an absolutely extraordinary animal. This is the animal. It's about that long, 2 inches long maybe. It has these extraordinary pectoral and pelvic fins that it can use to stay in place in the most rapidly moving water. It has no melanin pigmentation. It has no visible eyes. I've sectioned these fish. I can find actually no evidence in the adult of any eyes. It's extremely unusual for a cavefish. Most of them, even as adults, have small residual eyes. This shows the animal in nature. The other was in an ashtray in the lodge before we sacrificed it. This shows an animal in nature just maintaining its place in the thin stream of water that's flowing by. This is my student Luis again, the one setting the rope in Mexico, and here we're looking for Cryptotora in rapids. And this shows, without the soundtrack, it shows the animal holding its own in rapidly moving water. And this one, maybe the quality of the video isn't good, but it really does climb waterfalls. It's quite extraordinary. It has no competitors, and what it eats, we think, are just bacterial slime. So, switching continents, this is a cave catfish. It has tiny little eye spots. It doesn't respond to light behaviorally. You can flash a bright light on it, it doesn't respond to it. It's also very light in color. This guy, or female, this fish, is also about the size of the other one. This comes from Brazil. So I have good colleagues and good relationships with people in Brazil and Thailand and Mexico, and these are the places where I do my work. This shows another species, also Brazilian, not quite as light in color, filmed in the cave. The water it's dealing with isn't moving quite as quickly as the water that Cryptotora dealt with, but these guys are quite capable of exploiting the environment that they're found in and where they live, and have a characteristic look that all of us know denotes that they're cavefish. And then the final few examples, this is a karst formation in central Thailand. It's overlain by harder rock, which is how it's survived, whereas everything around it has been eroded away. And there are caves in this formation, and we've collected different species of cavefish there. Some of them are completely eyeless, or at least externally eyeless. Some of them of the same species have small eye spots, non-functional. But all these fish I've shown you may have a little bit of yellow coloration or pink coloration, but they tend to lack melanin, and they certainly lack a visual system. And that, of course, comes about from the environment in which we live, but there are questions about exactly what were the forces driving the evolution of this eyeless condition. And just to make the point that there are certain other interesting aspects of these caves. Lots of the caves in Thailand and some in Mexico have bad air, so you're breathing 4 or 5% carbon dioxide and existing on 17% oxygen, which is actually not so hard to do. It's a little bit painful, but if you're not exercising hard, it's okay. If, however, you repel into it, you really find it very, very difficult to climb out of it. And this is Jerry Campbell, who's in charge of caving safety assays, going the extra mile right before we enter one of these caves. Was that a grr I heard? I forgot for a moment who I was talking to. Well, that's Jerry's thing. Cavefish exhibit this convergence in loss of eyes, in loss of pigmentation, and in other characters. They've changed, it's not as obvious, but they've changed their metabolism. They're far more efficient because if there's not much food around, you really better be able to make the maximum use of what you can get. And I guess this will be the final final slide on convergence. So the bread and butter species, these Mexican cavefish, come from this area in Mexico and are found in, they're not all shown here, but 29 different caves in this area over 140 kilometers from here, the northernmost cave to the southernmost cave. They actually have derived from at least 2 separate species of surface fish that have been in the area. There was one set of surface fish that inhabited the streams around these caves and gave rise to populations over here. Then they went locally extinct. Another one came in and gave rise to populations here and there. So independent invasions. This is an old slide. I say 4 separate origins. We now know it's 5. Independent invasions, independent stocks, and yet they all converged on very much the same phenotype, the same look. One of the convergences, remarkably enough, is for sleep. So this is a record of activity of animals, of 4 different groups of animals, over a 2-day period. So the daylight, 12 hours, nighttime, 12 hours, the second day, 12 hours, second night, 12 hours. And what's plotted here is the amount of activity, the amount of inactivity, sleep, during 10-minute periods throughout this time period. And you can see that in black, surface fish sleep at night. Most of those 10 minutes at night are spent not moving, sleeping. Same for the second night here. They sleep much less during the day, although they seem to take Mexican fish, they seem to take a siesta around midday. And in colors, red, whatever this is, gold and blue, we have the same record, same type of record for three different species, three independent populations of cavefish. They all have very much reduced amount of sleep. Here is a bar graph. Surface fish sleep 800 minutes a day, and the three independently derived populations of cavefish sleep 200 or 100 minutes a day. We don't really know why this is the case. We think that if you're in an environment where there's not much food and you can't predict when the food will come by, you better be awake when it does come by in order to get it. But that's just a hypothesis. We haven't tested that yet. So it's not only eyes and pigmentation, it's metabolism, it's behavior, it's lots of things that have changed. Am I speaking too long? Okay. So as was shown in the previous graph, for sleep, we're looking at three different independently derived cave populations of the same species where you have convergence. And the convergence is shown here. Here's a fish from Molino Cave, northernmost of these Mexican caves that have cavefish. And then here's one from the Pachon population. These guys are albino, these guys are albino. These guys are completely blind, these guys are completely blind. They both have very small residual eyes I'm sorry, I'm searching for a term, but that'll do. Something deep in the head, and they certainly look very much alike. I can actually tell the difference between them when they're swimming around in the tank, but you probably would not be able to. So this convergence that I speak about is really very well exhibited by this group. And I think this is the point I made in the introduction. I meant to make it here. Convergence is to evolutionary biology as replication is to experimental science. 100 different species of cavefish is 100 replicates of the same experiment. One species with 29 populations, that's 29 replicates of the same experiment where because they all came from the same species of surface fish, you have a better idea of what their genetics is. So, Darwin. Darwin, when he wrote The Origin, knew about cavefish. He also knew about cave salamanders from Slovenia, but in the United States, there's a group of cavefish that had just been discovered, and obviously these were of great interest to him. He knew, as well as everybody did, everybody believed in evolution or change through time. He knew that they had descended from surface fish. And so he knew the polarity of the change, and what he's saying here, much more eloquently than I could say it, is if you poke my eyes out and put me in a cave, if you put me in a cave, I can't see. If you poke my eyes out, I can't fail to see any more efficiently than I failed to see when I had my eyes. It's dark in a cave. So he's saying, I can't see how eyes are injurious to animals living in darkness, and he knew nothing about genetics. He had actually been mailed a copy of the journal that had Mendel's paper in it, but he never actually read that paper. And so he simply had to attribute the loss wholly to disuse. We now know, of course, much more about genetics, so we know about genetics, and we know from Darwin about natural selection, and we know other means, not driven by natural selection, by which a character can, that isn't needed, can degenerate. And so there's some residual debate as to whether eyes are selected against in the dark or not having a function. There's natural selection will not be policing and screening new mutations that come along. So new mutations might come along, not be selected for, but just by chance increase in frequency in the population. And structure tends to run downhill if it's not tended. The metaphor I used before was the metaphor of the untended house. If I move away from my house for 20 years and nobody's watching it, I mean, I know for sure when I come back, there'll be window panes broken, there'll be leaks in the pipes, there'll be squirrels in the roof, there are squirrels in the roof. But what I don't know is where these window panes will be. I don't know which tiles or shingles will be pulled up by the squirrels and so forth and so on. So if natural selection is not policing a phenotype, keeping it up to snuff, keeping it up to par because it's needed, in this case because it's not needed, then things can go downhill. That's called genetic drift as opposed to natural selection. So major questions, I'm not sure I'll cover them directly, but if it is selection, how, referencing Darwin's dilemma, I don't really understand it, he said. Mutation, is it random mutation, is it drift? I've explained it all. And one of the things that we're interested in as well is how many genes have been changed between being an eyed surface animal and a cave fish. Is it one gene that's knocked out, or 2 genes, or 10 genes? Before we started this research, nobody actually knew. Now we know it's probably on the order of 10, 12 genes per population. So I haven't any memory of why I put this in here. I assume that I'm going to show you fish from Tinaja, Patron, and Molino, or something like that. Oh, yeah. So here's our surface fish. We know the polarity of change. Here's the Molino cave fish, which is an albino, as I mentioned. And here's one from a different cave, Tinaja cave. You can see a little bit of dark pigment in the mid-lateral line of that fish. These are actually not albinos, but they have greatly reduced numbers of melanophores, these pigment cells that are found in greater number in these guys. And when I hybridize them, what I obtain is a fish that in its pigmentation looks very much like the surface fish, maybe even darker than the surface fish, and very different than either of its parents. This is a phenomenon that's called complementation. And let me illustrate it with an example from human biology. Hearing disabled people tend to hang out with one another. I mean, they speak the same language. I can't do that, but they can understand one another with sign language. They tend to marry, as in all marriages, they tend to have children, and many times their children are also deaf. They're also hearing disabled. Sometimes, however, the child is able to hear. And that raises the question, is this guy the father of the child, or is somebody else the father of the child? And most of the time, he actually is the father of the child, 9 out of 10 times. And what it is she's deaf because gene A is bad. But gene B, which is also involved in hearing, is good. He's deaf because gene B is bad, but gene A is good. So he contributes in the sperm one good copy of A and a bad B. She contributes one good copy of B and a bad A. And the good copies of A and B are sufficient to make a hearing enabled child. And so that's complementation. The good qualities of one complement the bad of the other, and vice versa. And so here I have this diagrammatically for pigmentation. One of the fish, let's say Molino, has a good copy of A, but is homozygous. One copy from its mother, one copy from its father. Of B is little b, but it needs big B for dark pigmentation. It doesn't have it. Here, the fish from Tinaja is light in pigmentation. It has little a, and that's the deficiency. It needs big A, but it has big B. He contributes big A and little b. She contributes little a and big B, and the offspring is dark because it's complemented. That was a much simplified diagram. We now know that it's about a dozen factors in each population. But these dozen don't, I wish I had 3 hands, these dozen don't overlap with these dozen, and therefore you have this complementation. The complementation is also seen in vision. And so here's a 2-minute video illustrating this. Unfortunately, I can't play the soundtrack, but I can narrate it for you. The Mexican tetra has 2 forms. I feel like I'm dowsing. I'm looking for the microphone. But the purebred cavefish always are blind. When you hybridize them, however, you can get fish that can see. So what we did is we tested them in this little apparatus with this cup with white and black barrel staves. You put the embryo, because we worked on embryo on fry, you put the fry in the bottom, you whirl the cup. If the animal can see, its eyes follow the stripes and snap back, follow and snap back. So this is a surface fish, very small fish, big eyes, you can see them flicking, and in this bubble and in the shadow on the bottom you can see the movement flicks. This is a cavefish with small eyes, and you can see the bubble is, the reflection in the bubble is turning, the eyes aren't moving at all, it's really blind. This is another cavefish. You can see the movement over there, but the eyes aren't moving. So he's blind. Then I'm going to show a hybrid between those 2 cavefish forms, and you'll notice that the eye spots are much larger. So eye spots are larger, and you can see them flicking as the surface fish eyes flicked. So this is one generation of hybridization, one cave and another cave. Just as with pigmentation, these guys are blind because of changes in a certain set of genes. Those guys are blind because of changes in a different set of genes. In the hybrid, you get this complementation. So it's actually the same diagram that I showed before to explain restoration of pigmentation, except I changed this word to vision, and those are references to vision. So interestingly enough, when we published this story in Current Biology about 2 years ago, it was picked up by lots of news media. It was actually, for a brief time, on the most emailed science article list of the New York Times, but I was much interested in that it was picked up by Richard Dawkins. He really liked that story. And it was also picked up by a website called Creation Safaris. The creationists really like evolution in cavefish. Their argument is that only God can make something as complex as an eye, but random mutation can break down something that God has made and destroy it. So they're perfectly happy with the idea of regressive evolution. And in fact, their synopsis of my paper, I thought, was the best written and clearest of all the synopses that I read. It was really quite remarkable. And I know who I'm talking to, and I feel the same way myself, but it really quite amazed me. So, 3 conclusions, and I can shut up and take questions, or go. First, convergent populations or species can profitably be analyzed as replicates of evolutionary experiments. And I guess I haven't really shown that. It would be what I would talk about next if we had time for it, but maybe we don't. In cavefish, okay, this is the part that I haven't really explained to you. Direct selection is implicated in eye regression. And the third is that people can read into any story whatever they feel like. If you'll give me 3 more minutes, I will summarize what I would have shown you on those other slides.
Can you talk about what you mean by direct selection?
Okay, if you give me 3.5 minutes, I'll show you. So, if at any genetic locus you hybridize a cave fish with a surface fish, in the F1 hybrid you have heterozygote. It's cave surface. It's gotten one gene from one parent, another copy of the gene from another parent, and the other copies, we use the term alleles. The alleles are different. So gene has actually two meanings. One is a place on a chromosome, a genetic locus. So we might talk about the eye color gene. It's here in the genome. The hair color gene, it's there in the genome. But at that eye color locus, you can have a gene for blue eyes or brown eyes or whatever, and that second meaning of the word gene, we use allele for different alleles. Then if you breed an F1 with an F1, you get a 1 to 2 to 1 ratio of homozygote, heterozygote, homozygote. If you test enough genetic loci throughout the genome, you can use the information from that experiment to put together a picture of the genome. So here's one of the linkage groups or chromosomes in that genome, and here's a gene called OCA2, which is the gene for albinism we've been able to show in those fish. And here's something called 218E, which is just an arbitrary designation. All of those numbers are arbitrary designations for loci that we can demonstrate. The whole map would be far more extensive. It would have 25 of these linkage groups. We have 23. They have 25. And given the map like this, we can ask, does it matter at 105B whether an individual is cave-cave, cave-surface, or surface-surface? So is there a difference in eye size between those three? And most of the time, the answer is no. So when we look at gene 205E, the CC individuals, cave-cave at that locus, are medium, have medium-sized eyes. The heterozygotes have medium-sized eyes. The other homozygotes have medium-sized eyes. And what that means is that 205E has nothing to do with controlling the eye size differences. And it's not near anything in the genome that has anything to do with controlling those eye size differences. But when we look at 218E, these individuals in this class tend to have smaller eyes than those in the heterozygous class, which tend to have smaller eyes than those in the homozygous surface class. What that means is that near 218E, there's a gene that actually is controlling the development or the maintenance of the eyes. So something that's responsible. And so we use this sort of analysis to find about a dozen such places in the genome that really were important for eye size. And here's the answer to your question. When we did this analysis for eye size, every one of those places we found had the same polarity, in the sense that the surface-surface class, each one of these lines it denotes the trajectory for an individual gene. The surface-surface class was always bigger than the cave-cave class. But when we did the same analysis for the number of melanophores, we found that most of them were in, what we call the right direction, but some of them had exactly the opposite polarity. And the difference between this pattern and that pattern is statistically significant, and we replicated it, the importance of replication, we replicated it in an analysis of another cave population, so we're pretty sure that fate didn't play a trick on us. And the interpretation of this consistency is that natural selection is doing the job. Every time one of those mutations comes along, it increases the fitness of its holder, and it increases in frequency in the population. Whereas here, we view it as a case of genetic drift. If you don't have a melanophore cell there, you're going to have some other cell there. It really doesn't matter what cell you have there in your skin. If you're in the dark, you're not making melanin anyway, so it doesn't matter whether you have a melanophore cell there. There are several predictions that come from this. One of them is, since natural selection changes gene frequencies very quickly compared to genetic drift, we would predict that eye size decreases more rapidly in the cave population once it gets into a cave than the number of melanophores. We've looked at 6 other species of cavefish, and this prediction is borne out. The second part of the hypothesis is reason. Why should eyes be selected against in the dark? The brain is the most active tissue in your body, and the retina has a higher metabolic rate than any other part of the brain. It's an extremely expensive tissue. In us, 10% of our rods and cones are shed every day, so we renew the photoreceptor layer 36 times a year. Now, if you put me on limited rations and took out my eyes, the savings in energy probably wouldn't amount to very much, because I'm a mammal at a high basal metabolic rate. But fish, when they're not moving, have almost a 0 metabolic rate. And I wouldn't be surprised if the retina uses up maybe 1% or 2% of its daily energy budget. Testable hypothesis. I have a colleague in Scandinavia who's now doing those experiments on oxygen uptake of isolated retinas. So does that answer your question? And so I've spoken too long. I thank you for your patience, and I'll take your questions.
We were getting the mic up. Yeah, okay.
Okay.
You have to speak into the mic.
Eh?
Into the mic. Speak into the mic.
Yeah. I have 2 good questions. One is one of the mutants found eyeless, the eyeless gene of PAX6, I think it's called in vertebrates. That's one question. Second, as a fruit fly geneticist, I would like to ask if somebody has ever grown fruit flies for years and years and years in the dark. I suppose they have. I haven't looked at the literature. I suspect you have. And have they lost their eyes? Okay, that's
So,
2 questions.
So the first question was this master control gene, eyeless, it's called eyeless in Drosophila, PAX6 in other organisms. Has that been implicated? It was the first thing that we looked at. It has nothing to do with it, and these places during the or throughout the genome that we've identified, none of them are near where PAX6
Push power.
Wow.
Yeah.
Wow.
So, first question. Second question, I don't know about experiments looking for eyeless condition or inducing eyeless condition in Drosophila in the dark. It would be a great thing to do. One of the problems with doing an experiment like that is it takes 10 or 12 years, and you have to be publishing a couple of papers every year, and so that's something you do in your basement or not in your laboratory. Sorry.
Yes, is this working yet? What does this all mean to us ordinary people? I mean, how does it affect us humans? And what does it mean to us? Why should we pay attention to this?
Especially if creationists say God created the earth, and we atheists want to be able to say, well, we have important evolutionary evidence that maybe it's
Go
not
use the
so.
microphone.
Well, I'm not working for NYC atheists, and this is not my main consideration. I find creationists extremely irritating, and I've had requests for information from them. Stephen Jay Gould, before he died, I asked him about this. He says, just don't even deal with these guys. There's absolutely nothing you can say, and I think there is nothing that you can say that will convert them. They've been converted. To me, the most amazing thing is that with probably 5,000 religions on the surface of the earth, how people can believe they had the great luck to be born into the one true religion. So that's a different story. In terms of intellectual excitement, I think that's fun. And so that's the value that I get out of it, and I hope it's a value, and I know that it's a value that's imparted to kids, because I maintain this great fish room with 7 different species of blind cavefish in it, and everybody who visits the lab, they bring by their kids. They really love it. So I think that has a value in and of itself. And I don't have to actually listen to atheists' interpretation of it. I think, so for example, one of the things that I've discovered is, you know how they say a shark can detect a drop of blood in the water a mile away? I don't know whether that's true. I've never seen any experiment along those lines. I mean, does somebody go out and cut himself and wait? But I know that my fish, the surface fish, can detect amino acids, because I've done the experiment, amino acids in the water down to 10 to the minus 6 molar. I can translate that into real substances or real values that you can understand. My cave fish can detect amino acids in the water down to 10 to the minus 11 molar. So if you had 2 Olympic-sized swimming pools and you spat into one, and it was just pure amino, it's a vulgar illustration, but it makes a point, and it was pure amino acids in your saliva, and you stirred it, and then you took a cave fish and you dipped it into one and dipped it into the other pool, it could tell the difference. I don't believe that that's regressive in any way. That's a progressive change, and so I think there are elements of constructive evolution that the cave fish show. Finally, although it isn't what motivates me, the same genes that are causing loss of eyes in cave fish are the genes that are really necessary. There's no such thing as an eye loss gene. What there is the silencing or the corrupting of a gene that's necessary to build eyes or maintain eyes. Some of the phenotypes that we see are really reminiscent of human eye conditions like macular D, where we have a late age-related degeneration of the retina. And so if we could come up with a model for human eye diseases, I think that would be useful. Most of you are younger than I am, and some of you may actually be able to put your head down on the pillow at night and wake up 8 hours later. Don't work that way with me. I'm up 2 hours later worried about this, then I get back to sleep, and then I'm up at 4, I worry about that, and I get back to sleep. So the bouts of sleep that I have are many of short duration, and the difference in sleep patterns between the cave fish and the surface fish have to do with the number of bouts and duration. I know that NIH is very much interested in sleep disorders, so I think it's intellectually exciting, I think it has practical significance, and I'm really not out to try and convince people who believe otherwise that I'm right and they're wrong. Does that answer your question?
Yes, thank you.
Can I just to kind of summarize, would it possibly for you to just kind of take us through the journey, say, of a sighted fish, and just kind of, at least maybe just schematically plot out the point at which its descendants are eyeless. A sighted fish swims into a cave. 100,000 years later, its descendants have no eyes. Could you just kind of take us through that?
Okay, you just did.
Thank you very much. Always glad to help.
So,
my office hours are.
So there's this fish that shows up on our menu, tilapia. It's an African cichlid, and one of my ambitions is to die of old age without ever eating tilapia. About 25 years ago, the Department of Urban Ecology, I think the department has probably changed its name, in Mexico, decided that it had to feed the people. And what it did is it got large quantities of these tilapia fish and dumped them into streams and rivers all over Mexico. Amusingly enough, the fisheries department of the same government recognized that this was a form of pollution and would go out to these same streams and kill all the fish there to get rid of the tilapia. But the tilapia won. When I go into caves now, I find tilapia in the cave. That really means that if there's a cave and if there's a fish on the surface, within a short amount of time, that fish gets washed underground where either it can't survive or it can survive. If it can survive, then the population is maintained there. It's a radically different environment, and I really don't know the answer to the question, does it take 10,000 years, 100,000 years, a million years? I know it can't take 5 million years because the fish got into this area from South America only about 2.2 million years. I know that the process has run its course in all of these separate different populations. So if I imagined that it took a long time, it took these 2 million years in order to reach this end state, I would predict that I would go into one cave and they wouldn't be halfway there, and another one they'd be very much there, and it would be all over the map, but it's not over the map, so I'm guessing that it takes a short time. That wasn't short for the answer, but does that answer it?
That's fine. Just go ahead.
The question that I have is, it's not so much a question, it's that I think it might have been a little bit more appropriate to have identified a process which they only alluded to. And just said it plain directly. These fish have not lost something, they have simply shifted their major sense modality. And you pointed out how vastly more sensitive they are to taste, particularly of amino acids, whereas they've lost vision. And I think it just might have been appropriate to talk about evolution favors survival, and the modality that the fish will use over a period of millions of years will be the modality that most likely guarantees their survival in this particular case, taste over vision.
Right.
Yeah, that's doubtless true.
Chemical senses on this.
Yeah, but I think you're still fighting the creationists with that answer, and again, that's not my battle.
Welcome to New York City.
Thank you.
Can I ask you a question about plasticity? No. Presumably, once you get a loss of visual input into the brain, there are parts of the brain that are innervated normally by visual input that aren't anymore. Are those parts of the brain reprogrammed or used for anything else, or do we know yet?
Yeah, the optic nerve is smaller. The optic tectum is a little bit smaller. Nobody has done any electrophysiology that could tell you, because they're fish, that could tell you whether other senses are taking over. But that hypothesis actually dates back over 100 years to people who are just starting to think about that.
I think in the beginning of the talk you said that the second switch in the side switch that arrived a few million years ago.
I'm sorry.
Can't hear you.
I thought you said that the sided fish, the surface fish, and the cave fish diverged a few million years ago.
Yes. I don't know whether I said that. There are two stocks, and they seem to have diverged 6.7 million years ago. And I think that divergence between those two stocks probably occurred when they were in South America. One of those stocks gave rise to some of the cave fish, the other stock gave rise to the
But
other
I
cave
thought
fish.
you impressed the divergence over that amount of time meant they were different species and therefore could no longer mate and could no longer produce offspring.
Yeah. The question was, are we dealing with one species or more than one species? I refer to it as one species. Actually, probably it's two species. The old stock and the new stock are different.
But how can they hybridize them? How can they mate? I think that definition of the species meant they couldn't mate.
Yeah, that's actually wrong. Different species can mate with one another. It depends upon the group. With fishes, even things more distantly related than that can mate. I see abnormalities in the hybrids between stocks that are the two species. One of the abnormalities is that I look into the fish tank that may have 100 fish that came from a mating, and they're all female. So the sex ratio, the sex determination mechanism isn't working quite properly there. It's actually difficult for me to find males so that I can make an F2. The other thing is we've been looking at sperm motility, and we found that hybrids between different cave populations that probably are from these different stocks, that are from these different stocks and probably different species, the sperm has lower motility, so they're really less fertile.
Can you just tell me how the creationist critique was so clear and how it was so good, I guess. I don't know if it was good. We said the creationist critique of your paper was very
they wrote a very clear, I mean, they may be stupid, but they're not stupid. They wrote a very clear summary of what I had written.
not diluted, not just
Yes.
That's good.
Yes.
Hi. I would like to know, how in the world do you tell that a fish with no eyes is sleeping?
My friend.
There are in organisms where you can't do the electrophysiology, like in fish, you have to look at the behavior. And there are two tests that we've done. One is we keep them moving all night long, jiggling the tank in which they are. And when we do that, they sleep more the next day. That's called sleep rebound. If you keep me up at night, I'm going to sleep more when I next have a chance. The second thing is, when I'm asleep, I'm not listening as carefully or monitoring the environment as carefully. My thresholds for arousal are very much increased. And we can, by a simple assay of tapping on the side of the tank, when fish have been immobile for 1 minute or more, or less than 1 minute, determine that the threshold for arousal is greatly increased if they've been immobile for 1 minute or more. That's our definition of sleep. If they've been immobile for 1 minute or more, then we count them as sleeping. Frequently, those 1 minute or more actually are 60 minutes in length, but all they need to do is be immobile for 1 minute, and they're just not paying attention. Does that answer it?
Yes.
Your fish must really love you.
I put all of this into the hands of my graduate students.
There's a tendency for us to think that we are more evolved as a higher animal than fish. And I'm assuming that these fish are just as evolved as we are in terms of time. They're evolved. But it would strike me that as we get, if there is a higher form of evolution, does the speciesization become narrower margins? In other words, if a creature is in a more primitive state as it would be like several million years ago, would it be broader in its ability to, would it not have these narrow margins of speciesization?
I don't know whether this is something I can intelligently comment on, because it's not really an interest of mine. I know Stephen Jay Gould would talk about humans as an accidental presence in the age of bacteria. So he thinks that bacteria, or thought that bacteria were the dominant organisms and we're just, there. And he certainly was a powerful voice for the view that there's no such thing as evolutionary progress. I don't really believe that. I think that what humans do really is at a higher level, and our cognitive abilities are obviously much greater than other animals. We certainly have as long a lineage going back to that primal ooze that the fish do, but I think we've either been luckier or unlucky, or we've done something different with it. I can't really answer that question.
Just so I'm clear on how the evolution works with these fish, they live in dark caves, and they don't need to use their eyes much. So their eyes just kind of degenerate from disuse. But are you saying that they do improve a sense,
Chemical senses.
to keep them from bumping into the cave walls? How do they keep them from bumping into the cave walls?
I don't really believe, so no individual fish ever lost its eyes. It's a population that has a genetic change in it over time and evolves towards smaller and smaller eyes. In addition to having a good chemical sense, they have also a very good sense of vibration. They have little projections called neuromasts over their body, and the neuromasts can sense pressure, and they can sense vibration, thank you, and movement in the water. They actually are attracted to vibration. If you put a tuning fork or something that will vibrate into the water, the surface fish ignore it. The cave fish treat it as if it's the indication of some insect or something that they can eat. So they have other ways of, in quotes, seeing what's going on around them. They're supposed never to bump into the walls of the cave or the aquarium. My fish have not read this, and I see them occasionally bumping into the walls.
They didn't get that memo.
Yeah, they didn't get that memo.
One more question?
No.
Yeah.
Two, all right, John.
Is there a common methodology that these blind species utilize to find food, to target the food, and to ingest it?
So I don't really know the answer to that because I haven't looked at the feeding behavior of many species, maybe 10 out of the 100, but they rely heavily on the chemical sense. There's a small food item that aquarists give to their fishes. It's called a bloodworm. If you took a ballpoint pen and drew a thin line about 1/8 of an inch in length, that would be a bloodworm. A dried bloodworm. If I took one of those, took 1/3 of it, dropped it into a 30-gallon tank with my Thai fish, the balatorids, within about 30 seconds after a plume of water has hit them, they start searching the tank. It takes no more than another 30 seconds for them to find that food item. I think for the most part it's sense of smell, sense of vibration in the Mexican fish, because if you blind, and I don't do these experiments, but if you blind a surface fish, let it recover from its wounds, it's perfectly happy in the tank, it can't see, put a live food item into the tank, it may detect it when it's about 2 centimeters away. When it's about 1/2 centimeter away, whereas the cave fish detects it when it's about 2 or 2.5 centimeters away. So again, by the vibration.
By vibration. Thank you.
Joe?
Yeah, this is a simply technical question. You differentiated the fish, like either cave dweller or surface dweller, by their habitat, but am I right to assume that they were not necessarily the same breed of fish? Like the surface fish, is that a breed of fish, or that just happens to be the way that you describe them, or the way you differentiate them?
Oh,
So they could be like
the
different,
question
would they
was
be like tilapia or some more this?
the
or
use
something?
of language, surface fish, just really, I meant to denote the habitat.
Right, I
Anything
understand
that lived in a surface stream or river and is constantly exposed, 12 hours a day on average,
Right.
exposed to light, and cave fish as anything that was living in caves. There was a bit of imprecision in my use of the word of cave fish, because there are actually a lot of surface fish that get into caves. They're not permanent inhabitants necessarily, and they're not adapted to life in the cave. When I'm talking about cave fish, I'm talking about the ones that, when I showed that first slide and asked, does everybody know what the cave fish is and the surface fish? You all knew it, that's what I was referring to.
No, I do understand that, but I'm saying, like, when you say surface fish, they were not all the same breed of fish to begin with, right? There were different breeds of surface fish. Is that right?
For the experiments on the Mexican fish, they're all the same species of surface fish that I was using.
Oh, okay. Which species was that? Like which
It's
kind?
called Astyanax mexicanus. We biologists are not very imaginative.
But would you say that those were only those? But are there a lot of different species that drift into a cave, and do they see the same?
The question
exact
was,
behavior?
are there many species that drift into a cave? The answer is yes. You can find lots of things that are washed in. Most of the time, unless that cave is abnormal and has a tremendous amount of food in it, those surface fish that are washed into the cave look really very skinny, and they eventually become fish food for the cave fish.
So she's asking, why is it that particular breed was able to
Right,
adapt as the others were not?
So the question is, why was that particular species able to run with it when they got in the cave? And nobody really knows the answer to that, But what is known is they don't need the visual sense to mate. The males and the females make pheromones. They actually mate in the dark, and if you blind them, they can mate successfully. So the sine qua non of maintaining a population is being able to find the opposite sex and mate with it, and they're able to do that in the dark, and maybe that other surface fish, other species
They
can't.
call that a blind date.
Thank you very much, thank you. It was great.
Hold on, give me a name. Give me a name.
Despite appearances to the contrary, this is not election night in Honduras.
Yeah, that doesn't make sense.
Bad joke. Okay. Luckily, the show's coming to an end. Our guest has been Richard Borowski, who is a professor of biology. We've been discussing blind cave fish. And you've been watching New York City 80s live on tape. I've been your host, Dennis Horvitz. Thanks for watching. See you again next week.